So in this session we will discuss the last minute revision part of biotechnology and its principles. Let's start the session with question number 1. Some multinational companies and other organizations are using bioresources for commercial benefits without proper authentication and compensation to concerned authorities. And you have to answer part A, give the term for this unauthorized act, and part B, suggest any 2 ways to get rid of this. So let's see the answer.
1. By appearance, benefits of bioresources should be shared between developed and developing nations. Laws should be developed to prevent unauthorized exploitation of the bioresources.
क्वेश्चन नंबर 2 बहुत इंपॉर्टेंट है, बहुत अच्छे से देखिएगा इस क्वेश्चन को. A bacterium Bacillusthuringiensis produces a toxic protein named cryprotein that is lethal to certain insects but not to bacterium. So there are 3 parts in the question. Part A, why this toxin does not kill the bacteria. Part B, what type of changes occur in the gut of insects on consuming this protein. And part C, how man has exploited this protein for his benefit. अगर आप लोग को आंसर्स पता हैं तो well and good, नहीं पता है तो let's see the answer.
2. Produced in an active form as prototoxins. Prototoxin becomes active toxin in alkaline pH of gut of insects. Toxins bind to surface of midgut and cause perforation, swelling, lysis of cells, ultimately leading to death. See specific Bt toxin genes isolated from Bacillusthuringiensis and incorporated into several crop plants such as cotton and corn, which become pest resistant against certain insects.
In question number 3, how does DNA recombinant technology help in detecting the presence of mutated genes in cancer patients? बहुत इंपॉर्टेंट है बच्चों, आंसर्स पता है तो वेल एंड गुड, बिकॉज दिस इज द लास्ट मिनट रिवीजन सेशन मेनली फॉर द क्वेश्चंस व्हिच यू हैव टू बी प्रैक्टिस्ड. So let's see the answer first.
3. A single-stranded DNA or RNA tagged with a radioactive molecule is allowed to hybridise with its complementary DNA in a clone of cells followed by detectin using autoradiography.
ये क्वेश्चन नंबर 4 जो आप देख रहे हैं ये काफी बार पूछा गया बोर्ड एग्जामिनेशन में. नेम द सोर्स ऑर्गेनिज्म ऑफ टैग पॉलीमरेज and what is the specific role of this enzyme in PCR. PCR stands for polymerase chain reaction. So if you know the answer, well and good. Otherwise, let's see the answer.
T. aquaticus is a bacterium that lives in hot springs and hydrothermal vents, and Taq polymerase was identified as an enzyme able to withstand the protein denaturing conditions high temperature required during PCR. Therefore, it replaced the DNA polymerase from E. coli, originally used in PCR.
In question number 5th, name the selectable marker in the cloning vector PBR322. Mention the role they play. बहुत इंपॉर्टेंट है बच्चों, रोल ऑफ PBR322 एज वेल एज आप सारे वैक्टर्स बहुत अच्छे से पढ़ के जाएं अपने बोर्ड एग्जामिनेशन के लिए. चलिए आंसर देखते हैं इसका.
In the cloning vector PBR322, ampicillin and tetracycline resistance genes are the selectable markers. They also help distinguish recombinant cells from non-recombinant cells.
So let's see question number 6. It is very important. Write the palindromic nucleotide for the following DNA segment. Segment आपको पार्ट A and B के बीच में दिख रहा है. सेम पार्ट A भी है. Write the palindromic nucleotide sequence for the following DNA segment. It's 5 to 3 prime GAATTC. And its part B is name the restriction endonuclease that recognizes the sequence. Let's see the answer.
A5GAATTC33CTTAAG5. B. EcoRI is the restriction endonuclease that recognizes this palindrome.
क्वेश्चन नंबर 7 के चांसेस बहुत हैं पूछे जाने की, because 7th part is very important. Mention the number of primers required in each cycle of polymerase chain reaction. Write the role of primers and DNA polymerase in PCR. One more part is there, that is part B. Give the characteristics, feature and source organism of the DNA polymerase in PCR. तो चलिए, अगर आपको आंसर्स पता है, well and good. Otherwise, let's see the answers of these questions.
Each cycle of PCR requires 2 sets of primers. Role of primers: primers are complementary to the regions of DNA. They anneal to both the strands of DNA, which are then extended using nucleotides. B. DNA polymerase is thermostable. Source is Thermus aquaticus. The separated DNA fragments can be visualized only after staining the DNA with a compound known as ethidium bromide followed by exposure to UV radiation. The tumor-inducing plasmid T can be used as a useful cloning vector in gene cloning in plants. It introduces a piece of DNA which transforms normal plant cells into tumor cells. The T plasmid contains genes for the synthesis of authentication, cytokinin hormone, and its introduction in a plant helps it to produce its own nutrient. Machinery.
तो ये सारे क्वेश्चन इस चैप्टर्स के बहुत इंपॉर्टेंट है, एक बार प्रैक्टिस जरूर करके जाएं एंड ऑल द बेस्ट फॉर द बोर्ड एग्जाम.